13 different beaks. Advantages that made them tigers, wax into.
A lot of times you'll see marine iguanas shoot a salt spray.
No other cormorant in the world is.
Too many things I have to set up. Okay, is that coming through? Is that loud enough? All right. So what we need to do today is to finish this video, which is only about 10 more minutes, I think. We're going to talk about the 2 videos afterwards. We'll have a little discussion about the videos. And then I need to go back and do something from chapters 15 that I forgot to talk to you about, okay? And then that should take up maybe half hour, 45 minutes. And then we'll have some time for questions, okay? All right, so let me start this.
The blue-footed booby does a special mating dance called sky pointing, supplemented by honks and whistles.
Loud enough?
Special mating behavior and colorful displays might be a way of helping creatures recognize and mate with others of their own species. A definition of species being a population that reproduces only within itself, preserving its distinct identity.
This is Black Turtle Cove on the north coast of Santa Cruz Island. These sea turtles come into these protected waters to mate. The male green turtle is smaller than the female adult green turtle. It's advantageous for the female to be larger than the male because that way her buoyancy can support the male and keep them both above water. She could actually drown. This is Roca Redonda, one of the most dramatic seabird concentrations in the Galapagos. A hardcore bird watcher would just go nuts here. Really love it. Thousands and thousands of seabirds.
To share the view of the great Galapagos seabirds, the Discovery expedition brought along its own airplane packed in crates for assembly on deck. Called an ultralight, this is a small, open, 2-seat flying machine powered by a snowmobile engine. were off limits to tourists, and of course they were unattainable for Darwin. They seemed to fulfill every fantasy of fascinating lost islands in the Pacific, craters still fuming, scenes untouched by time, no sign of human presence. The Galapagos seems a perennial source of new things, said Darwin. Today's scientists will second that. They will also tell you that new things, not just new creatures and vegetation, but new bacteria, viruses, all forms of life, are not only evolving, but evolving in many cases much faster than Darwin thought. And it's happening everywhere. The difference between evolution in these islands and elsewhere is only that nature's creativity stands out more vividly against the pristine landscape of the Galapagos.
I think I'm not recording.
Very rewarding. We've seen something new every day, something new to us, something new to science. Yet we will have to spend a lot of time verifying and confirming that what we saw has not been overlooked or misidentified or misinterpreted in the past in the literature. I'm sure that much of what we do over time will be challenged and questioned, and that's all part of the process of science.
6 months after it ended, John McCosker estimated that the expedition had discovered about 24 new species, meaning that a discovery took place on almost every day of the diving. Two scorpionfish found by the expedition are not only new species, but belong to no known genus. A new genus will be created to demonstrate their evolutionary uniqueness. Classification of new species is a long process. It might take several years. Other important findings will be evaluated over even longer periods as the academic discipline of a lab follows up on the excitement and adventure of discovery.
We think that this is probably a new species that's not been described before, and I'm certain that this is the only footage available of cirrate octopuses in good condition like this.
We don't know what species this is. We're not familiar with it. It might be unique to the Galapagos. It'll take a lot of work in the museum examining other specimens and comparing it to be sure of its identity.
This is Colomonima, a beautiful little deep-sea medusa that has some very special tricks up its sleeve. Its color pattern in white light is one of blue tentacles with white tips and a bluish tint to the swimming bell. Also, when white light shines through the muscle tissue that comprise the bell, the fibrous structure of the muscle tissue breaks up light into shimmering patterns of iridescence.
This creature is a batfish. His public fins are modified into fins which can walk, very large eyes, seen as prey, and between his eyes is a little organ used to emit chemicals which attract smaller vertebrates to his mouth.
Pelagothurian is completely unique among the sea cucumbers in that it never spends any time on the sea floor. This is the first time that anybody has had the opportunity
to see what it feeds on and to be able to characterize its role in the deep sea community here. This is an extraordinary deep sea fish. Common name is the tube-eye. This is probably the first living specimen anybody has ever had in a position to be able to photograph.
And while the fish people study their specimens in the future, Dr. Dent will be sorting and studying 15,000 bones brought back from Barn Owl Cave. David Stegman excavated bones representing at least 12 species, 9 of which are extinct on Floriana Island, and 2 of which were found for the first time. Meanwhile, further study of stratifications in Barn Owl Cave revealed prehistoric changes in climate with an impact on evolution.
Galapagos is such a great place to study evolution. And evolution is really the key to life. Evolution as time perspective to life. We can look at where we came from, where other organisms came from, and those same processes are going on. It gives us a peek at the future. In some cases, a really good look at the future. It's sort of ironic we're losing a lot of things right now when we're finally getting the tools necessary to study it in enough detail to understand it.
Okay. All right. So. Right. Okay, so let's talk about these 2 videos. And I've got the sheets for the questions on them if anybody wants to look at them. Why did I have you do this? Why did we spend class time? I think I told you the very first day of class. Professors covet class time. We don't do anything in class that we don't think is valuable. So why did I think spending parts of 3, was it 3 or maybe 4 class periods watching these videos? Why would I give up that much time? Okay, it relates to the topic we're talking about and it'll be on the exam, that's true. But I could have lectured. Okay, it's applications of what we're talking about. Anything else? Okay, puts it right there smack in front of you. It looks at adaptations, and instead of me just telling you about it, you can actually see things and how they relate to the organisms that we're talking about. Anything else?
You also made a statement that you said you wanted to give us a break from lectures.
Yeah, give you a little bit of a break. I hope watching a video is a little, I mean, they have a lot more production value than I have. So hopefully that was a little bit more entertaining in the process. And I think the photography, especially in this one, not so much in the other one, but the photography in this one is just fabulous. The things you can see. Okay, let's talk a little bit about some things that the 2 videos had in common, and I'm going to try and lead you to a conclusion, okay? In the part of the first video where they were in the rainforest and they were collecting samples of everything, trying to identify all the species in the rainforest, okay, think about that segment. Think about the segment, the last segment in that one where they were regrowing the rainforest, no, the tropical dry forest. Okay, think about that segment. Think about the part here where they were at the Darwin Center, where they were protecting the baby tortoises. What can you see in those three particular things, scenarios, I guess? What did they have in common?
They're trying to protect species.
Trying to protect species.
Humans were the one that messed it up. Like in the first place.
Okay, so humans messed it up, so humans are trying to make things better. What else did those three particular examples have in common? Can you think of anything else they had in common? Who was doing the work and directing it and all that kind of stuff? What? Scientists were. But was it just Americans and Europeans coming in and saying, do it this way?
Everybody working together.
One of the big things I think that I saw in those three particular examples, and also I think about this until a second, the example when they were out on the ship and they saw the fishing boat illegally fishing too close to the shore, is it was the native peoples in that area that were doing some of the work and and providing leadership, as well as the scientists who were coming in from out of country. And I think that's a really, really, really important thing that sometimes Americans lose sight of. And that is that when we go in and say, do it this way because we know more than you do, is that a very effective approach? Absolutely not. When we go in and say, tell us what you want us to help you do, we can provide some funding, we can provide some expertise, but this is your project. And I think that's one of the big things at that Darwin Center, that the native peoples there had control over it and did it themselves with perhaps some input of expertise and input of money from outside sources. Because I think way too often there's this tendency for the big bad Americans to come in and tell other people what to do. And people don't like that. And I don't blame them. I wouldn't like it either. I don't like it when somebody comes in and tells me that I'm doing everything wrong and that I should do something different. And if you think about ways that we could potentially save some of these ecosystems, it's not going to be from the top down in all likelihood, unfortunately. It's going to have to be buy-in from the people who live there. Okay, what can regular folks do that would be helpful to these ecosystems? What can we do? Is there anything we can do? Okay, we can pick up trash.
Raise awareness of how much we need them.
I'm sorry?
Raise awareness of how much we need them for the environment.
Okay, raise awareness. What else can we do? Try again.
Trying to prevent burning my forest species.
Okay, that's a really big one. Trying to avoid introduced species. How many of you have ever gone somewhere and had gone camping or gone hiking or gone to somebody's house and had a bonfire or something, and you take wood from where you live to where that site is? It's a very common practice. It's a really, really, really bad thing to do because that spreads pests. That's one of the reasons the pine beetle, is that what it's called, in the ash borer, the emerald ash borer, has spread. Part of it is climate change because they can now survive the winters further north than they used to be able to. So a lot of pine forests have been almost wiped out by the pine beetle. And part of it is because people are taking firewood from where they live or where they have a farm they can get wood from to another place to burn for a campfire. And while that sounds like an okay thing to do, it's cheaper probably, it carries with it whatever pests, whatever insects are on the wood. Now you can say, well, we're burning the wood. Aren't we burning up the insects? No, because they will crawl out of the wood when the wood starts getting hot. And that is a huge problem. And I know that's been an issue for quite some time because my kids are 28 and 31. And it was a problem when they were in Boy Scouts. And the troop that they belonged to was big on camping and had a lot of people, leadership in it that were like skilled workers, carpenters, plumbers, you know, people who had very specific skills. One of the reasons we chose that particular troop. And they did not understand why we were telling them, stop taking wood from your farm and taking it to wherever you're going camping, because that's a dangerous thing to do. And they didn't get it. So there are things like not taking introduced species other places. And that's a simple thing we can do. It may cost us a few dollars to buy local firewood. Okay, but it could prevent the spread of an introduced species that's very damaging. What else are things we can do? Think about the rainforest. What's happening in the rainforest, the tropical rainforest? They're cutting down trees. What are they doing with the trees? I mean, partly they're cutting down the trees so they can build factories and farms and stuff. But what are they doing with some of that wood that they're not burning? They're turning it into furniture and other products. So what can we do that would not encourage that? Don't buy furniture that's made of those woods. Okay? What about nuts? Do trees have to be alive to produce nuts? Yeah, so eating nuts might be a good thing to do. Although I will tell you that almonds, how many of you drink almond milk? It is not milk, okay? It is not milk. You cannot milk an almond. It is terrible. Have you read the ingredients?
I don't know how to make it.
Have you read the ingredients that's in almond milk? Bull. It's not real. It is not natural at all. But where are most almonds grown? California. What do almond trees need to make almonds? Lots and lots and lots and lots of water. What does California not have a whole lot of? Water. So what is happening in California? They are using tremendous amounts of water for almond trees. Is that a good use of water? No, not really. Not really. And it's contributing to the fact that almost the entire state of California, it's not, but the entire state of California is covered with smoke, I can tell you that. My future daughter-in-law was just in Montana. Montana? Yeah, the Glacier Lake National Park, I think that's in Montana, right? Told you geography is not my thing. But anyway, it was smoky there because of all the fires. And so using water for things like growing almonds in California is contributing to that problem. So we can make a choice whether or not to do that. Now, it takes water to make regular milk too. But you don't have to graze cows in places where there's not water. What are other things we can do? What can we do that would be useful? Recycle. Unfortunately, what's happened with our recycling lately? Recycling has virtually come to a standstill because of political issues. There's no market for the recyclable materials anymore. So the only thing that's really getting recycled much is aluminum, steel, cans. Cans are still getting recycled. some bottles are getting recycled. That's about it. Paper's not much getting recycled. Landfills. How many of you know the problems in landfills?
They make greenhouse gases.
They give off greenhouse gases. They also take up a lot of space. And things don't decompose in a landfill. Back in the 70s or 80s, they did this, and people there was no recycling much then, okay? Everything went in the landfill. And this group did a study of landfills where they took like core samples. They had a machine that would go down in a landfill, and then you could tell by how deep it was in the landfill how old the stuff was. And what they found was things don't decompose in a landfill much. They found steak that was in the landfill that still was identifiable as steak, like 10 or 15 years later. Lettuce. Newspaper just sits there. That's one way they could date stuff, because they got newspapers. And I was so excited. My kids were born in 89 and 92. And I had a friend from Pennsylvania who was using cloth diapers instead of disposable diapers. And I tried it, and I just hated using the cloth diapers. And it wasn't because of washing them, although I will admit that's a yucky job. But it was because when I would change the disposable diaper, the kid's skin would be dry after just a pee diaper, okay? With a cloth diaper, there would be little dots of urine sitting on their skin. And I hated that. Okay? So when they did this study, and I read about it, it was great because disposable diapers were not a big problem in landfills. The things that were a big problem in landfills were construction waste. Did you think about that? Every time you make something new, what was there, all that material goes to the landfill. Newspapers are a huge problem. And food. Those were the big problems in landfills. And they could correlate certain things. There was a beef shortage in the 70s, I think it was. There was a beef shortage. And there was more beef waste in the landfills during the beef shortage than there was before or after. Because people were hoarding. Guess what? Americans are hoarders. Americans are horrible people. What happened when this whole pandemic started? People started hoarding things. Toilet paper. I couldn't find oil, flour, cornmeal. I'm like, seriously? People are hoarding flour? And there were a couple things I just really could not figure out why people were hoarding. Okay? It was amazing, the things that were not available. And you're horrible people. And people did the same thing with beef back in when there was a crisis of shortage of beef. And then people ended up throwing it away because they had more than they could use. So that's a problem. And then people started selling it on Amazon or eBay or somewhere like that. So, because a few things that I would normally buy that weren't available in the store, I looked on Amazon or eBay, and normally I'd pay $8, and they're on sale for $45 or $90. It's like, I don't need it that bad. So, anyway, those are problems. But there are things we can do, and we all make those choices, and we all decide what's more important to us. When I was your age or younger, I saw a documentary about children making Oriental rugs who were like chained to these looms. And so I decided then and there I would never own one unless I knew the origin of that rug. I would never own one, and there is not one in my house. And my in-laws had hundreds of them, and I won't even take them. Okay? Because it's a decision I made as a teenager. Some people are very concerned about coffee and fair trade coffee and chocolate and stuff like that. And so they adjust their buying to reflect that. I don't drink coffee myself, but my husband does, so we try to buy the free trade coffee. So we all make those choices, and you will face those choices as well as you move along. Do you have any reflection back to me, any comments, any questions about the videos that we watched? Anything you want to comment on? Did you like them? All right, so the next thing I want to do is go back a little bit to chapter 15. Okay, here. All right, screen share. All right, so what we need to look at in chapter 15 that I kind of forgot to do was the origin of eukaryotic cells and the origin of multicellular organisms. I realized it when I was going through putting together the study guide. I was like, oh yeah, I forgot to do that. Okay, so where did eukaryotic cells come from? We think there were two successive processes. In one process, the eukaryotic cell's endomembrane system formed. Because what's the definition of a eukaryotic cell? What does a eukaryotic cell have that a prokaryotic cell doesn't have? A nucleus, membrane-bound organelles, right? So what they think happened to make the nucleus and the other portions of the endomembrane system is that prokaryotic cells, which at this time, at least some of them, didn't have a cell wall, and something occurred that basically caused what we call an invagination of the cell membrane. In other words, the cell membrane kind of folds in on itself. Remember, if you think of a cell membrane as like a balloon, it'd be like you took your hand and you poked it in the balloon and then you kind of pinched it off. Okay? And so if you do that, think about the nature of that membrane. That means the inside layer of the membrane would be like the outside membrane of the cell, and the outside part of the membrane would be like the inside of the membrane, of the bilayer, okay? So this part on the outside would be like the inside, and the inside would be like the outside, because that's what happens when you poke your hand in and then you pinch it off. And we find that most of the endomembrane system is encased in kind of a double membrane, which makes sense again. If you're pushing something in and pinching it off, you're going to end up with a double layer of membrane. And so we end up with cells that encase the nucleus in that double membrane. And there are intermediate forms. There are organisms that have sort of nucleus, but not quite all the way to a nucleus, if that makes sense. So that's the first process, is the formation of internal membranes, basically. The second process is called endosymbiosis, and that is a word you should learn. Endo means what? Inside. And symbiosis, what do you think of when you hear symbiotic? Some sort of relationship, okay? So what they think happened here was that very early eukaryotic cells, I think the other process started first, the formation of the internal membrane. These cells would ingest, basically, an aerobic heterotrophic prokaryote. Aerobic meaning it's using oxygen. Heterotrophic meaning it's not doing photosynthesis, it's getting its food elsewhere, and it's a prokaryote. And these, instead of destroying this, some of them occasionally, again, we're we're talking over millions and millions and millions of years, occasionally would survive the process of being ingested. Okay? And they would then set up a symbiotic relationship where the big cell provides protection for the little cell and provides raw materials, and the little cell provides bunches and bunches of ATP. Okay? Same thing with some photosynthetic prokaryotes. They could do photosynthesis. They were ingested, and perhaps a few of them survived instead of being completely destroyed, and they lived in symbiosis and became chloroplasts. And there's actually at least two, they think, in evolutionary history, at least two separate events that led to development of chloroplasts. There's a fair amount of evidence for both of these. For example, mitochondria and chloroplasts have their own DNA. And that DNA looks like bacterial DNA. It doesn't look like it. In other words, it's circular and there's one instead of being linear like our chromosomes in our nucleus are linear and they come in pairs. Okay, so that's one example of how they are very much like prokaryotes. Interestingly, mitochondria, you get all of your mitochondria from your biological mother, not from your biological father, because basically your biological father is a DNA donor. Okay? DNA enters the egg cell and that's about it. You may get a little mitochondria, but not much for sure. And it turns out that you can do genetic pedigrees using maternal mitochondrial DNA. I am the end of the line for my mitochondrial DNA, or really I guess my sons are. I have no daughters. I have no sisters. My mother had no sisters that had living children. My grandmother, my maternal grandmother, had no sisters that had living children. And I think her mother, my great-grandmother, had no sisters that had living children to pass on their mitochondrial DNA. So you have to go back several generations before there would be somebody who had the same line of mitochondrial DNA. And by that many generations, every generation there's going to be some mutations. Okay? So my sons will not pass on their mitochondrial DNA to their children. Excuse me. Their children will have their mother's mitochondrial DNA. So it is a way that they determined that a couple of young women, or they were probably older by then, by the time they figured this out, in Europe who claimed to be Anastasia. Remember the Tsar Nicholas, and at some point in time they thought Anastasia might have escaped from the execution, and a couple of women in Europe claimed to be Anastasia, and they were able, because of the way the royal pedigree goes, everybody knows who married who and who had children with whom, so they could look at that mitochondrial DNA, and they determined that none of the women who claimed it were actually Anastasia. None of them were the daughter of Alexandra. So, for males, you can follow a line using the Y chromosome. I think we already talked about that. But women, you can follow a line with mitochondrial DNA. So here's an example of an aerobic prokaryote. This would be like something similar to the precursor of mitochondria. This would be a photosynthetic prokaryote, which has structures in it that are very much like the thylakoid membrane. Okay, we're not going to go through the diversity of protists. We talked a little bit about this in lab. Looked at. Okay, the question then is, where did multicellular organisms come from? how did we get from unicellular organisms to multicellular organisms? The big difference is not just that they have more cells, it's that they have specialized cells. Cells that are specialized for a particular function is really what you have to have before you have true multicellularity. You can have a colony of cells, and we saw this in lab. Remember, we saw the Volvox were a colony, the Anabina, remember, was a long line. That was actually prokaryotic cells. But you can have a colony of a unicellular organism. At some point, when you start getting some specialization of function, you're kind of on the border between unicellularity and multicellularity. In the Anabina, we saw a few cells that were bigger, and those were called heterocysts that were specialized for reproduction. Okay. So that happens. Once you start to get several different kinds of cells, particularly if they start making gametes, then we consider this to be a multicellular organism. So we move from unicellular to a colony to sort of a borderline to what is then truly a multicellular organism. And we do this with both plants, animals, fungi, which are the three most common, or the three kingdoms of multicellular eukaryotes. All right, so that's really what I wanted to pick up then from chapter 15, because that is fair game for this exam. So has anyone looked at the study guide? I posted it last night, or late yesterday afternoon. Chapters 13, 14, and 15 that were on the quiz 2, that material is fair game for this exam, but the exam will mostly be 15, these last couple things I just talked about in chapter 15, and then 16 through 20. So kind of like exam 1 had a little bit from quiz 1, but was mostly after quiz 1. This will be similar. Okay? All right. Yes, ma'am. My preference would be to have the exam over the weekend online, and then we could use Monday for a review session. That would be my preference. Some of you indicated you'd rather have an exam in class. But then I mean, there's just no time. We don't have a reading day. There's not really a time to do that I can do a review session that everybody can come. I don't think. I kind of looked. I sent you all the final exam schedule, and it's just boom, boom, boom. Almost every hour.
We should do it over the weekend and then
I'm sorry.
I think it would just make more sense to do it over the weekend, that way we can review everything.
Yeah, that way we can have time on. We can have a good amount of time on Monday, and just we can go all the way back to the beginning, and then we can review, kind of, to set you up for success on Wednesday.
Sorry, did you have it on the weekend? Like, I know, like, Nick or work all day on the Saturday and Sunday, so like, would you have it open for the weekend, or like, certain hours, or what?
What I did last time, I think, was I opened it Friday evening, and then it was open all weekend.
It was.
Yeah,
Okay, so it'd be available, we could do it anytime during the week.
yes. Isn't that the way, I think that's the way I've done it.
Yeah, you could do stuff for like the children.
Yeah. It'll be, do you prefer it on Canvas, or did you like it when it was partly on Mastering Biology and partly on Canvas? You like your Canvas better?
Biology was really slow, it kept messing up.
Yeah, that's the thing with Mastering Biology, I know it lags a lot. Did you like being able to look at all the questions?
Yeah.
Okay, because that's more like taking it in
I
class.
also like that you didn't have to walk in your question immediately.
Didn't have to what?
You didn't have to like walk in your question immediately, so like if you saw another question.
Yeah, you could go
basically
back.
gave your answer,
Yeah,
you could go
That way, when you could see all the questions, you could go back and change an answer if you wanted to.
You know how long is it gonna be?
How long it's gonna be?
Yeah.
Would I give you 35 minutes for the quiz? Probably give you an hour, 55 minutes to an hour or something.
How many questions will it be?
I don't know. Some of the questions, I will tell you right now, will come straight from the video. So if I were you, I would have those video sheets right in front of me.
Like, these videos are like.
And then there, make sure you, I mean, some of you are not doing the, Canvas, the lecture stuff that's on Canvas that I posted, the chapters 13, 14, 11, 12, and 11 and 12 can be on the final exam. Okay? Some of you are not doing those assignments, I don't know what to tell you. You have to do them. It's part of the course. So do them. You can't just do the mastering biology and not do the lectures, and I can tell who does the lectures.
What mastering biology be factored into our final grade?
Mastering biology is worth 10%. The way I have it set up is 1,000 points, okay? And so I will look at your mastering biology. The first thing I look at is, did you do all of the mastering biology? Did you do it? Number 2, I will look at how often did I have to give you extra time to do it. Number 3, I will look at how much time did you spend on it. So people who did the mastering biology, an assignment that's supposed to take an hour and 20 minutes, and you did it in 15 minutes, that's going to ding you, okay? Because that means you're not watching the videos, you're not doing the exercises. You're just answering questions, okay? And then 4th, I will look at your score. That's like the lowest thing that I look at. And then from that, I will make a judgment out of 100 points of how much you got. I will say most people get at least a 90, if you do it.
Yeah.
If you don't do it, you're not going to get that many points. It's free points. I mean, I don't know what to tell you. So that's how that will factor in.
Will you put that in like sometime next week?
Yes. You will have until the final exam. If you do it after the final exam, you're going to get 0 credit for it. What is the point of doing the homework? It's to help you do better on the exams. That's the reason I require it. From my perspective, it's also to help you learn the material, okay? Because I think if you learn the material, you'll do better on the exams, okay? But from your perspective, the point of doing it is to help you do better on the exams. If you're not going to do it, it's not going to help you on the exams. There's no point in doing it after the fact. So you'll get a ding if you do it after the exam that it covered as well. That's the reality of that. In terms of plants and animals, I would go ahead and look at those lectures that were online. I would go ahead and look at the mastering biology, not mastering biology, you already have to do that. The PowerPoints, I would at least flip through them. Especially on the plants and animals.
So is it more helpful to know like videos that work, like you explain in the PowerPoint or the PowerPoint? Like for those lectures?
Like for the plants and animals, the videos that were in there. Well, I told you you didn't have to spend a whole lot of time on those. I just wanted you to get the big picture. And it showed lots and lots of video of animals moving and that kind of stuff, which I thought was cool. But I didn't really expect you, I mean, each of those videos was like 10 to 15 minutes long, weren't they? So I didn't expect you to sit there for 4 hours and watch videos, unless you just enjoyed it. Plus, you can watch those YouTube videos, you can watch it double speed.
I have a little question about the plant life cycle and the spermatocyte
Okay.
and whatever. I'm getting very
Okay, so who asked me about the US? Okay, so the first thing to understand about plants is that they do this alternation of generations. They have a multicellular haploid and a multicellular diploid. Animals only have a multicellular diploid. The only time animals have a haploid is the gamete itself, but plants are different. So in moss, for example, you have this green leafy stuff, and then you have these structures that grow up out of the moss. And I looked for 2 hours, and I had my work study students look, and we could not find moss with sporophytes. It's just the wrong time of year. Okay, and but these sporophytes are 2 in. Okay, and these sporophytes are the zygotes, grow from the zygotes through the process of mitosis and make the sporophyte. Then the sporophytes make the spores, which then undergo meiosis to make, and the spores then are 1 in. The spores undergo mitosis and grow into the gametophyte. The gametophytes undergo mitosis and make the gametes. Then fertilization takes place, and we get a zygote, and we're back to the diploid. And the weird thing about bryophytes is that what we think of as the plant is the haploid stage, is the gametophyte. So the way it's been explained to me, and I'm not a plant person, okay? The gametophyte makes the gametes, okay? The sporophytes make the spores.
So the spores go through meiosis, or does the sporophyte go through meiosis?
The spores what?
Does the spore itself go through meiosis, or the sporophyte?
The sporophyte undergoes meiosis, and the spore itself is haploid. So both the spore and the gametes are haploid. Only the sporophyte, well, the zygote, which then grows into the sporophyte, is diploid in a plant cell. And the different thing about moss is that what we think of as the green leafy part of the plant is the haploid stage, is the gametophyte. Until I started teaching here, I didn't know those words. Okay, so. Now, in the other plants, when we move to then what are called the ferns and the fern allies, here then the sporophyte, that's the moss, then the sporophyte then is what we think of as the fern. And the gametophyte are those little things on the back of the fern that we call sori, that then grow into a new plant. So it looks like it really only gives you this particular, it only really gives you the life cycle here. The big thing I want you to know is that plants do alternation of generation. They have a multicellular haploid and a multicellular diploid state. Okay? The multicellular haploid state is called the gametophyte. The multicellular diploid state is called the sporophyte. And for most plants, what you see as the plant is the sporophyte, except for moss and bryophytes. When the part of the plant you think of as the plant is the gametophyte. Seeds contain little plants. The seed, when you cut open a seed, is considered to contain an embryonic plant. It's already diploid. And you'll get a lot of this in 214 next year. Good question. Anyone else have a question about the material? Yes?
Are introduced species
say, invasive and
Introduce, are they the same thing?
more or less, invasive is kind of the next step beyond introduced. Introduced is a new species in an area. It becomes invasive if it kind of takes out other. But in a lot of senses, they're used interchangeably. Just invasive is the result of introduced. Dandelions are not native to the United States.
Are you gonna make a study guide for the final exam or will you?
The final exam, the study guide for the final exam is just put together the study guides for the individual exams.
Okay.
What I expect on the final exam will be less detail. Okay? It will cover the same material as, all the tests, but I will go in less depth. What I think I told you at the very beginning, one way to study is to think about it in layers. Think about when you make an outline and you have your Roman numeral 1, 2, and 3, and then under Roman numeral 1 you have capital A, B, and C. And then under capital A, B, and C, you might have 1, 2, 3. And under 1, 2, 3, you might have little a, B, C. And that's just going down into more and more and more and more detail. Well, whereas on an exam, I might expect you to go down to 1, 2, 3. On the final exam, I would probably only expect you to go down to the A, B, C. Does that make sense? Not as deep. This is a survey course, so we don't go super deep. You probably went, if you took a good high school biology course, you probably went into depth more on several topics than we did. But now we have 3 more classes where we'll go into depth. In 212, we'll go into depth on the first third or half of the semester. And then in 214, you go into depth in plants and animals and a little bit of fungus. And then in ecology and evolution, you go into depth on the last part of the chapter, last part of the course. So this is an overall course to get everybody kind of up to speed, hopefully. Other questions? Anything else? So what's your task between now and Sunday? Okay, before you take the exam, what do you need to do?
Mastering biology.
Do your mastering biology. What should you do before you do the mastering biology if you haven't already done it? Watch the videos. Okay? Yes?
I just have one little to say. So like in your, I don't remember what video. Okay, the ecology video. So you talked about this author named like Rachel Carson. Over books like this morning. Yeah, I kind of found the answer review this morning and I opened and who is the author of Rachel Carson and it was, I don't remember what it was. So I was like, there's no way I'm gonna tell Dr. Hinton how my foundation I can read this because this is
It probably was from Silent Spring.
It was. They talked about something about it, but it was something about how like she didn't think that you could use pesticides and stuff because she called them biocides, like it affects the rest of everything else.
Yeah. Yeah. She wrote other books that were wonderful. She wrote several children's books that were beautiful.
They said that she won like all these like awards for her books, and I was like, no, Dr. Hinton said they're awards.
Her one book is boring, Silent Spring. If you try and sit down and read the whole book, hopefully the section they pulled out for you to read was interesting.
Yeah, it was.
But that doesn't mean the whole book was interesting. I never had to read it either. When you have to read something, it's usually less interesting than when you get to read something. But I actually didn't have to read it. It was just one of those things. If you're going to be a toxicologist, kind of should have read, Silent Spring. It's kind of a understood thing that you're supposed to do, and I just found it incredible. But I like to read murder mysteries. So. It's not nearly as interesting.
Murder mysteries.
Murder
How
mysteries.
did you think you like majored in?
What?
Toxicology, the only thing you like majored in or whatever?
That's what my graduate degree, my master's and doctorate are in is toxicology. Sort of pharmacology of toxicology. Okay, any other questions? All right, I will put up the exam sometime, hopefully this afternoon. I'll let you know when I put